Hello, everyone. I'm Frederic Runge. I'm a PhD student at the Machine Learning Lab in Freiburg, and I'm very excited to present our recent work, Partial RNA Design, here at ISMB. Partial RNA Design is a joint work together with my colleagues Jörg Franke, Daniel Pferdmann, Rolf Backhofen, and Frank Hutter. So let's start. Oh, that's strange. Okay, when we talk about RNA design, what we typically mean is that the ultimate goal is to design RNA sequences that fulfill certain functions. And this can be broken down into two parts. On the one hand, we have our desired functions, which are typically connected to a certain application we have in mind. For example, we might want to regulate a certain gene, and we can do that, for example, by designing a riboswitch construct that triggers activation of the gene. Or we might want to design some genetic circuits, and we can do that by using toehold switches, for example, that can be used to define logic gates. And also the development of mRNA vaccines can be translated into a design problem where we might want to optimize the transcription or translation rates of our vaccines. And there are many more applications. But on the other hand, of course, there's this process of designing RNA sequences where we typically start from a certain design goal. And this is strongly connected to our application. So, for example, we might want to have a sequence at the end that folds into a certain structure. Then, of course, we need to define some constructs. We need to define properties that we would like to have, or define the structure that we would like to have at the end. And then we can start to search for promising candidates. And these candidates then have to be analyzed. This can be done in a wet lab, for example. And unfortunately, with our first iteration, we typically do not end up with the desired results. And there is often a refinement step where we have to restart our cycle again, starting with the design goals, refining them, or refine our constructs. Now, computational RNA design means that we would like to develop a computational method that helps us to automate the process of finding promising candidates. And to do so, we typically translate the design goal into a certain objective that we would like to optimize. We can use the defined constructs or define constructs to define a search space that helps us to guide our search. And finally, of course, we need a design algorithm to find promising candidates. Now we can have a look at a simple example here where we'd like to design RNAs with a secondary structure that contains a certain tetraloop motif, GAAA here in this case, as depicted here at the top. Below this small figure, you see the dot bracket notation, string notation for the structure, where dot corresponds to an unpaired nucleotide and a pair of matching brackets corresponds to a pair of nucleotides in our structure. And then we can choose from different algorithms here to apply to this task. For example, we might want to start with a sequence-based search approach. We can simply define a sequence search space here, and then the EF approach would be to assign random nucleotides to the mass positions. Of course, this is arguably inefficient, but we can also use, for example, a sequence recovery objective to train a model on a large set of sequences that already contain our motif. Unfortunately, of course, this requires that we have previously known sequences, a large set of them to train our model, which might not be available for all the different design tasks if we start something new. So maybe it's better to use a structure-based design approach here, where we define a complete construct at the beginning, and then we can, for example, use an MFE defect objective where we use a folding algorithm to fold our design candidates and just compare them to the desired structure we have. But again, we see whenever we start refining our construct, we of course have to define the entire construct again and then rerun our algorithm. So maybe it's not the best solution here. And we might end up with better results using a shape-based design approach where we just define the shape of our desired target, a single hairpin in this case here. And using sophisticated methods, we can also include sequence constraints and just define an objective that the folding of our design sequences should have a certain shape. Unfortunately, again, we run into the problem that whenever we change our construct at the beginning, we have to rerun our algorithm. So our observation is that there is currently no general approach available to solve our task satisfactorily. And a solution would be to have a new flexible RNA design problem formulation, as well as an algorithm, of course, that can design RNAs directly from the provided motifs that have variable lengths and different composition. And that brings us to our contributions. We propose partial RNA design, which is a novel RNA design paradigm that considers any objectives. This problem is formulated as a constraint satisfaction problem, and it allows to define arbitrary sequence and secondary structure motifs that can be used to define a search space. And secondly, we propose libLearner, which is a new algorithm that can design RNAs directly from the provided motifs, considering varying objectives. And libLearner strongly builds on our previous approach, Learner, which is an automated reinforcement learning approach, which we proposed back then in 2019. So let's start with partial RNA design. And I would like to use the same example here as before, designing RNAs with a secondary structure that contains our tetraloop motif. So when we would like to define a search space here, our idea is very simple. We just divide the search space into motifs, and each motif consists of a secondary and a sequence part, secondary structure part and a sequence part. So when we look at our loop motif here, we can simply define the secondary structure part and the sequence part. Here you see that we also include a wildcard symbol, this question mark here, that corresponds to a position where we can assign any nucleotide to. And more formally, our structure space is just a sequence of structure symbols, and the sequence space is a sequence of nucleotides, including the wildcard symbol here, with a specific length of 6. And that's what we call a motif, defined by a sequence and a secondary structure part. Now, of course, we can add another motif for our search. As we have seen before, we might want to have this motif to have a certain structure as well. The main change is that we now have a length of 11. But unfortunately, we would like to search over multiple different structures here in this motif, as we have seen before. So our idea, again, is quite simple. We just include another wildcard for unknown positions in the structure space as well, as we have already done in the sequence space. And now we can define a motif just with question marks here to search over both of these structures at the same time. Unfortunately, again, we would like to search probably over more different constructs here with different lengths and composition, and not being limited to a length of 11 in this motif. And so our idea was, again, very simple, to just include another wildcard symbol here, which we call a variable length wildcard, which actually defines a region that is unconstrained and of arbitrary length. So we can then just include that into our search space formulation. And our motif now becomes a single symbol here, the single wildcard symbol, where we just say, okay, at this point, you can just give me anything you want with any composition. And of course, we can also do that on the other side of our loop region. And then our search space translates to a sequence of motifs, M1, M2, M3. 2 of them are defined by this variable length wildcards only. Now, when we search in this search space, we simply use 2 variables that define the sequence and the structure part of each motif, and we assign values to these variables simultaneously. Here are our motifs now, M1, M2, M3, and we just use these 2 variables, and then we can assign values to these variables. Our variables X and Y are defined as just as sequences of nucleotides and structure symbols. And finally, these variables will define our set of variables for the constraint satisfaction problem. And the X and dy will be their corresponding domains of values in this problem formulation. Now, the problem is, without any restriction, we can just assign any values to these variables. As shown here in red, we can assign a sequence that has a different length than our structure, which might not be the best outcome of this assignment here, because it will give invalid results here. And this is where our objectives come into play that help us to define valid assignments. For example, we can again use a sequence-based design approach, where we simply define an objective that whenever we have a constrained position in our search space, we would like to have this position assigned with the same nucleotide in our assignment for variables. However, this doesn't solve our problem, because actually X1 is not constrained in any way. So this is still a valid assignment. But we can also include a structure-based design objective here, where we just say we take a folding algorithm, use a concatenation of X1 to X3, and this has to fold into our design structure here. And this actually would solve our problem, because obviously our sequence won't fold into the same structure that is defined by Y1 and Y2 and Y3. And of course, we can do the same for the shape-based design, where we simply take, again, the folding algorithm and check for a desired shape. And finally, these objectives will formulate our set of constraints in the constraint satisfaction problem, which we look at now. So formally, this problem is defined as given a sequence of RNA motifs, in a set of objectives, the solution to the partial RNA design problem is actually a solution to the constraint satisfaction problem defined by our variables in the set X, their corresponding domains of values in the set D, and a set of constraints that are defined by our objectives. So this formulation has certain advantages. First of all, partial RNA design allows to precisely define a space of potential solutions via the objectives. And secondly, there is actually no restriction on the definition of the motive. So we use a full structure where we know about the exact pairing scheme of each nucleotide. But in this case, we don't need it. We can just define any construct or any motive we would like to have, and we don't need to define an entire construct. Now, looking at the algorithm we developed to solve this task, I would like to start with a certain observation, and that is that whenever we have a task that contains a variable length wildcard, we can actually interpret that as a set of fixed length tasks. And this means that we reduce the complexity of the problem a lot, because we can now consider only sets of fixed length tasks when we sample from this provided search space. Our approach to design sequences now is that we use a deep reinforcement learning agent that just fills the mask positions in the sequence. And then we use a folding algorithm to get the structure and train with with an adapted MFE defect objective here, which I will explain in a minute. And just to mention this once here, so the general approach is an automated reinforcement learning approach, which means that we use a strong Bayesian optimizer called Bob that jointly optimizes the hyperparameters, the neural architecture, and the training data for our approach. So at the end, this is a push-a-button technology where you just start the optimization, and at the end, you get a trained and optimized reinforcement learning system to solve the problem. Now, the training data we use is we just fold all sequences from the AirFarm database using RNA fold, and we mask parts of the sequence and the structure using a certain strategy described in the paper in more detail. During evaluation, we then allow updates of our policy network of the agent to being able to adapt to certain tasks or even to changes in the objective as defined by the problem formulation. Now let's have a look how we generate sequences with LibLearner. As I said, this is a reinforcement learning system where we consider an interaction between an environment and an agent. And this is done via states, rewards, and actions. So the environment first computes a state representation, which is communicated to the agent. The agent plays an action, which is fed back into the environment. And then the environment computes a reward signal to communicate the value of the chosen action back to the agent. Now for LibLearner, we start from a certain search space here of a mask sequence part and a mask structure. And we first define a task, and this task is used to define local views as the states. More precisely, we take the two representations for the sequence and the structure, we encode it into a joint representation, and then we pad this representation to define our task. And then we look at the mask positions of our sequence space to define a local view of this position. And this defines our state, and the state is then fed into the agent. The agent embeds the state representation into a certain dimensionality and plays an action G in this case. And then we can transition to the next site, which is masked in our sequence space, and just do the same thing again, while for this position, we actually ignore the sites that are already assigned values to, like the C and G. And we can do that for all the mask positions now to get a final prediction with our agent. And this prediction is then fed into back into the environment, which uses RNA fold to compute the structure of the design sequence. And then we compare the structure to our initial input structure space, comparing W hat and or omega hat and Y in this case, to see if the structure we have produced now is within our desired search space. And we, at this point, simply use the Hamming distance while ignoring masked sites of the predicted structure. This defines our reward, and we use that reward to update our agent. So this is how we train this thing. And now coming to the experiments, we have a lot of experiments in our paper. At first, we show that actually our strategy works well. We show that we can navigate all these large spaces that are partially restricted, but of a fixed length. And we also show that we can adapt, or the learner can adapt to different changes of the objective by just changing the reward. Finally, we show some task where we try to solve this partial RNA design problem. There we actually do very aggressive changes to the reward function, such that it doesn't have to do anything with the training objective we used, like there's no folding involved. For example, we design RNAs for a given mRNA target where we just directly optimize the free energy of the interactions. But I would like to highlight one experiment here, which I think shows best how we actually can apply partial RNA design and LibLearner to different problems, which is a design of TFLN riboswitches following a previously proposed protocol by Wachsmuth et al. I would like to note at the end that we do not retrain LibLearner for any of these experiments. So we just use the pre-trained model. We can change the objective, and through our adapting reinforcement learning system, we can just solve tasks with different objectives here. So for the design of TFLN riboswitches, the task is to design 50,000 riboswitch-like constructs of different lengths that can be used for subsequent analysis. And defining the search space, we follow Wachsmuth et al., starting from the aptamer sequence and structure here. We then would like to have a spacer regions of 6 to 20 nucleotides shown here. This translates into 3 motifs in our case, a motive to ensure the length of 6 in the center, and then flanked by 2 variable length motifs, which basically mark points of extension for our design. And then we have a 10 to 21 nucleotide region, which should be complementary to the 3' end of the aptamer, also with a position for extension. And then at the end, there should be a 8U stretch, which is mainly unpaired. For the objectives, we first start with our training objective, which is just this adapted MFE objective. But we also do experiments where we just add an additional GC content deviation objective to our reward function. As I said, this is just plugged in. We do not optimize for anything here or change any parameters. For evaluation, we first check that the construct has a 2 hairpin shape. There should be no pairing within the last 7 nucleotides of the 8U stretch. And we use a simulation of co-transcription and folding, also following Wachsmuth et al., where we just take a folding trajectory with an elongation speed of 5, and then check that there is no interaction between the aptamer and the spacer region in this simulation. So the results look quite promising. We see that LibLearner can actually produce much more valid candidates, so valid in the sense that they satisfy all these evaluation criteria compared to the original procedure. And we also see that we have a very high structure diversity here. When we look at more details regarding the length of the sequences designed, we see that LibLearner actually designs candidates that are quite uniformly distributed across all the different sequence lengths. And finally, for our experiment where we just add the GC content reward signal, we see a similar result where we see that whenever, no matter what kind of or what value we would like to have in the GC content, we can still design a lot of valid candidates here. So in conclusion, I propose partial RNA design, which is a new general and flexible RNA design paradigm. And we propose an algorithm, LibLearner, which can design RNAs directly from the provided motifs using varying objectives. And yes. So thanks for your attention. Please visit our paper. The source code is publicly available. We also have a small web server, which is still under development, but I just put it here. And to conclude this talk, I would like to end with this running example of the tetraloop motif design here, where we run a small example with learner about 1 minute to design about 50 different constructs here that all contain this tetraloop motif. So thanks again for your attention.
So great work. So does this work when we have pseudoknots?
So great question. Well, in the search space definition, actually, you can define pseudoknots, but you can actually use a folding algorithm that can produce pseudoknotted structures and just compare with this structure. So in principle, you can just apply any folding algorithm. So there is a difference between you have a certain search space definition that helps to guide the algorithm predictions, but the objective is not connected to the search space. So you can just define any objective, and the algorithm will just optimize for this objective. So if you have a certain structure that you'd like to have, you can use a folding algorithm to fold our structure or the sequences we have designed with this folding algorithm, and then compare to the desired pseudoknotted structure, if you like.
So I guess currently RNA fold is not capable of predicting pseudoknots, right?
RNA fold is not, but this is we mainly use RNA fold as our oracle during training. So when we for experiments, we also just don't care about folding at all and directly optimize for example, for the interaction energy with another mRNA target. But we can still provide different search spaces. So this is one experiment in our paper where we actually change the search space definition and just use this direct optimization of energies for RNA interactions.
No, if I want if I am looking for a structure which contains a pseudoknot, so this will produce those structures which contain the pseudoknot, right?
If you define the reward function that takes your folding algorithm that can produce pseudoknotted structures, you can actually compare these results with your pseudoknotted structure as well. Yeah.